Thank you, everyone, for coming today. I appreciate it. My name is Michael Pratt. I manage the Technology Development Office at Boston University. We do the patents and licensing. And, we do this once a year just to step back, take a break from all our busy work and just recognize the important work of some of our faculty. And it's not really part of the day job to innovate and to create patents and create new companies. And it takes that extra bit of effort. And I think it's worth recognizing that. So with that, I'm going to turn it right over. Melinda Shockley, if you can go to the next slide, Rose. Melinda Shockley is going to manage the panel, moderate the panel today. So I'm going to hand it off to Melinda. And then after the panel, Ken will come up and present the award to Wilson and say a few words, and then Wilson may say a few words as well. So Melinda, why don't you come on up? Melinda's our Executive Director of Faculty Entrepreneurship.
Thank you, Mike, and welcome, everyone. It's fantastic to be able to celebrate innovation here at Boston University. And it's especially exciting to be able to celebrate the contributions that Professor Wilson Wong has made to the fields of bioengineering and synthetic biology. So joining us today, we have several other leaders in the fields of bioengineering and synthetic biology. I'd like to welcome Jim Collins, Mo Khalil, Mark Grinstaff, and Justin Latendra to this event. Each will be presenting a 5-minute lightning talk, a bit about their role in this space and some thoughts in terms of developments that have been in progress. And then we'll follow that by a Q&A session. So I'd like to first welcome...
Okay, here we go. Hi, everyone. It's really great to be here. My name is Mo Khalil. I see a lot of familiar faces in the room, but just in case you don't know me, I'm now professor of bioengineering and molecular and cell biology at Harvard. But for the last 13 years, and this is a recent move, I've had the privilege of running a research lab right here in this building, right alongside Wilson. In fact, we really just came up together here. And as Jim just mentioned, there's going to be a lot of sort of similarities to Jim's talk here. BU has a storied history in synthetic biology, as Jim just mentioned, and it's been just like an absolute honor and privilege with Wilson to have contributed a little bit to that storied history and all the growth in synthetic biology here at BU. There's a little bit of memory lane here. I pulled up some pictures from various BU articles of sort of the various kind of variants of synthetic biology groups that have been established here. And I really like this photo here. This is actually back in 2012, I think, Wilson. This I think, photo was taken after both of us had our first lecture. So right after finishing up teaching class together. So it just gives you a sense of sort of the history and all the amazing things that we've been able to do together. Okay. It's, I think, defining synthetic biology is a little bit dangerous, and analogies are deceiving, but I kind of like this one, and I think it's quite apt. As many of you know, traditional biology has really been a study of using sort of observation and cataloging and characterizing things like, the parts, right? Genetic and molecular components of living cells. Synthetic biology sort of flips the script a little bit and asks, how do these parts work together? So if we could reassemble them into things like quote-unquote genetic circuits, as Jim mentioned, use those and embed those into living systems like cells and study how they execute function, we might be able to learn something about biology. And then, of course, we may be able to reprogram living systems like cells to do useful things. So many of you are very familiar, of course, with reprogramming microbes to produce useful chemicals. What I'm going to tell you a little bit about is some of our work in 2 other application spaces, applying synthetic biology in the field of medicine, as well as more recent work in trying to sort of advance agriculture. So it was actually Wilson, as Jim mentioned, that really got me interested, and our lab interested in this problem of the CAR T cell and immunotherapy. And this is this revolutionary sort of new paradigm in medicine where you can take, for example, a patient's own immune system, immune cells like T cells, and basically genetically engineer them to express these receptors called chimeric antigen receptors that redirect them to tumors and unleash the immune system. And because my lab was right next to Wilson, and he got me very excited, we got to team up on some of this stuff similar to sort of what Jim mentioned. And the idea was really quite simple. This is a revolutionary new medicine, but has its challenges. So what if we could implement genetic circuits where you could now, control these cells and like launch controlled therapeutic responses? And so we've had a lot of fun, working on this. More recently, we've been thinking about extending, cellular therapies beyond cancer and into chronic diseases. So imagine, for example, a living cellular pharmacy in an implant device that's producing, for example, drugs for type 2 diabetes and obesity. I want to also just tell you about some more recent work, which is thinking not just about the cell as a medicine, but cells as actually discovery engines for medicines, and in particular biologics. So there are these proteins called membrane proteins, like GPCRs. They're really important, and they're amazing drug targets, but they're actually notoriously challenging to study. So we have, over the last several years, thought about using yeast, which gives us our beer, bread, and wine, and reprogramming it to effectively display these types of targets and use it as basically a search engine for discovering antibodies that can target these. We were fortunate to take this technology and work with the OTD office here to launch a company called Ira Bio that's in South San Francisco now, using this technology to make antibody-based medicines. I promised to tell you a little bit about agriculture. I'll be brief. We face a major crisis, right? Our agricultural systems are under major pressure because we're going to have a massive population. How are we going to feed this population? Also, we have climate change, and this is sort of, really sort of affecting crop yields. And so we and others have been thinking about how we might apply our, technologies in synthetic biology in the realm of plants to try to, help create solutions for this. And just, one interesting project that has sort of emerged from this in collaboration with Mary Gearing's lab is try to develop a system for fixing hybrid vigor, basically trying to expand the benefits of hybrid crops, more broadly. And then finally, I'll end on this concept, just like Jim did, on this idea of taking synthetic biology and expanding it. How do we build much more complicated systems like that we see in nature? And this is taking folks into this realm of what we'll call generative biology, using massive data sets in a way, generating and using massive data sets to train models that can predict new biomolecules, new cells, new living systems entirely, and try to build them and use them in new applications. So with that, I'll just say, Wilson, congratulations. It's really great to be here, and thank you all.
So it's a pleasure to be here, and I want to say thank you very much to Wilson. I'm new to the collaboration. I've only been working with him for a few years, but that picture says a lot. He's always smiling, and it's really great to work with him and his students. And this collaboration really came out because of two students that were working in our labs, Josh McGee and Jack Kirsch. And we got interested in messenger RNA. So a single strand of RNA has a 5' UTR, has the next sequence of RNA that encodes for the protein of interest, and then our 3' UTR. And of course, all of us are very familiar with that because of the COVID pandemic, and where our, quote, vaccine was a single strand of messenger RNA, and in the center was encoded for the spike protein of the virus. And of course, we got an intramuscular injection. Now, the key technology that made that all work was the Nobel Prize-winning discovery by Carrico and Weissman about using modified bases into that messenger RNA strand. And of course, this got injected into billions of people and was quite a lifesaver. Now, there are two faults of this technology. One is the amount of protein that you express is actually fairly short, or a small amount. And the duration by which it expresses that protein is also fairly small. And so there are ways that one can think about improving this. And one of them is to use something called self-amplifying RNA. So the same messenger RNA sequence, but instead, in addition to the cargo, in this case the spike protein, upstream of that you have an enzyme which makes more of the RNA. So now when it goes in the cell, it makes more RNA, which makes more protein. So you get a lot more protein and you get it for a longer time period, which is really cool. The problem, this is really big. And your cells recognize it and degrade it so it doesn't work as well as you want. And what the two students did was this really cool experiment to actually look at different modified bases to see if they could regain function in a system that wasn't functional. And so what we found is that, I should say what they found, is that there were about 3 or 4 of these nucleotides that were actually transcriptionally and translationally active. This was really cool. So that means that we could actually deliver this RNA to the cell. The cell would basically not degrade it. It would allow for increased expression and for a longer time period. And so working with Florian, who was also on that first slide, we developed a COVID vaccine, all right? And at a very, very, very small dose, in this case 10 nanograms, the modified self-amplifying RNA, which is in purple here, basically was protective to about 75% of the animals. The wild type about 50%. Messenger RNA, which would be the equivalent to our Moderna and Pfizer vaccines, about 25 or so, and PBS not very protective at all. And so this technology opens up the idea of actually making low-dose, potent vaccines for all of us, and that's really quite remarkable. Well, as we know, Wilson likes logic, and so he's like, Mark, we should make something that has logic. And he says, well, can we make a piece of RNA that just basically expresses in a tumor and not everywhere else? And can we design a system to do that? And that's what Wilson did. And so the idea was, again, to build in some logic into this messenger RNA sequence such that it would not, for example, express in immune cells or in the spleen. And thereby, maybe after an intramuscular injection, you would actually see protein being expressed in the tumor and not in the liver or the spleen. And you can think about localizing your treatment to a particular tissue. And really, that's what Wilson's all about. It's all about design, engineering the system, and getting it to work just right. So Wilson, congratulations. Thank you so much. It's been a pleasure.
Okay. Hey everybody, my name is Justin Latender, and I was actually a graduate student in Wilson's lab for about 5.5 years, and the last time I was standing at this podium was my defense about 4 years ago. So super excited to be back, and 4. Yeah, is time flying? You're going too slow. But yeah, super excited to be back here talking to Wilson's accomplishments and to you all today. The focus of my talk is actually a little bit different from all of the other talks. I wanted to focus a little bit more on what it takes maybe to translate some of these applications of synthetic biology into the clinic, and maybe geared towards a little bit more of the student population here. So synthetic biology is really not a brand new field, but synthetic biology aims to solve heterogeneous problems through engineering control mechanisms. A couple examples of these problems include microbial consortiums for maybe re-engineering the gut microbiome, trying to target antigens on the surface of cells who maybe have very heterogeneous expression levels of those, and then maybe trying to express things in cells using endogenous cues by cell type or cell state, where, again, they vary by cell type and cell state. So just a quick highlight of an example of this technology. This is some work that I did in Wilson's lab, trying to improve a synthetic receptor signaling mechanism. And as you can see here, between cells receiving a signal and cells not receiving a signal, there is a difference, but it's very difficult to draw a clean line separating those two signals, and also there's a very broad response profile. So what we did is we tried to digitize it, again, using well-characterized parts, put in a couple feedback mechanisms, and at the end of the day, we were able to improve the signal-to-noise ratio of this system about 3 decibels. And so what I want to focus on today is how do we take these engineering principles of precision and control and use them to help us translate some of that type of technology and the type of technology that Wilson is focused on into drugs and drug development. So something that has really stood out to me in the literature are a couple of papers published by AstraZeneca over the last couple of years on using this 5R framework for translating synthetic biology, or really any biological advancement. The first point is choosing the right target. And breaking that down a little bit, what that means is, what does the drug do? Does it do what you intend to do? Is it affecting the population of cells you are intending to affect? And if it's not, think about killing it early to save money, time, resources. The second R is focusing on the right tissue. So eventually, the goal is to get these drugs into people. They're probably going to be dosed systemically. And again, in an in vivo model that replicates, hopefully, human biology, is the drug making it to the right area of the body, the right cell type, the right tissue, and is it acting accordingly? The third R is focusing on the right safety profile. And I think a lot of the time in academics, especially myself, when I was, developing in my PhD, I was not necessarily focused on safety implications of the technology that I was developing. It was a lot of the times more just, what is the coolest thing we can build? How efficacious is it? So really highlighting, are you using convincing preclinical models to validate the system that you're working in, and again, kill it early if it's not going to be safe or effective. The last two, I think, are a little bit further down in the pipeline, and maybe not as applicable to direct translation of these technologies, but eventually you're going to need to get these drugs into the right patient. So thinking about biomarkers that you can stratify your patient populations into to make sure that, again, you're treating patients who are going to benefit from the technology that you're developing, and maybe not give hope to patients who are not going to benefit. And finally, you want to make sure that your drug has the right commercial potential. So, how crowded is the space, and do you believe that the drug you're developing is actually going to be either first in class and provide hope to patients who don't have another option, or best in class to maybe do better than what's already available? And so taking that all into account, in a retrospective paper, AstraZeneca found that the system works. About 4 years after they published the initial 5R paper, they did a retrospective analysis, and they saw that through preclinical development all the way through phase 3 trial successes or failures, they had about a 5x average increase in success rate of different projects that they were launching from phase to phase, stage to stage, versus the industry standard, about 20% versus 4 to 5%. And I think importantly, through preclinical phase 1 and phase 2 development, looking on the right side of the screen, there is a significant decrease in failure rate among different projects that they were initiating because they were killing those projects early on based on safety and efficacy data that they were collecting through just discovery work that they were doing on molecules. And to highlight a quote that has stuck with me since my time in Wilson's lab, you want to fail quickly so that you can succeed faster. And I think that's exactly what these guys were setting up. To get to the point of drug development, you need to be able to launch what you're doing. You need to be able to translate it into a company or a patent that you can license. And I think that BU especially is a really great hub for progressing academic synthetic biology into the clinic. On this slide, I've just highlighted a couple examples of platforms started by folks at this school, either professors or students. And again, today is about Wilson celebrating his accomplishments in the space. Wilson and the Wong Lab have been involved in 3 startups, which I think is super impressive. And the goal here, again, is to translate synthetic biology into the clinic to benefit patients. Focusing on one that Jim had highlighted a little bit earlier, Senti Biosciences, they're developing some CAR NK technology that is pretty revolutionary and, again, makes use of circuit technology that comes directly out of Wilson's lab. On the left side of the screen, super exciting, is a paper published earlier this year by Sung Yi and a bunch of other colleagues on the knock-gate inhibitory CAR, which provides a mechanism to spare healthy cells that might be expressing antigens that are expressed on the tumor cells that you're trying to kill. So again, translating this to patient benefit, 50% of patients in an ongoing phase 1 clinical trial, data was shared at the end of 2025, benefited from this technology. And 40% showed a complete response. At the end of the day, that's all we're trying to do, is to benefit patients with revolutionary technologies that everyone in this room is working on. So with that, I want to wrap up and say congratulations to Wilson. It was a lot of fun working in your lab. I learned a lot, and yeah, excited to be here celebrating with everybody today.
I'll invite the panel up. Doesn't matter where you sit. Thank you for those wonderful lightning talks. It's just amazing, the potential, the promise of the field of synthetic biology and the successes that we've seen so far. And I'd like to thank Wilson, not only for your creativity, but for your commitment for translating innovation into impact. That's so important. The discovery is important, but the fact that you are now taking these learnings and translating them into products that can benefit humans. So thank you very much, and congratulations. So we're going to turn now. Our panelists just gave a wonderful whirlwind overview of work that they've accomplished, work that Wilson's accomplished, and a lot of the, again, promise and potential of the field. But I wanted to give them a little more time to expand upon some of the topics that they raised through some Q&A. Some of these questions you'll be familiar with, and you've already touched upon some of these topics in the various talks. I'm not going to call on each one of you. I mean, I'll start, but the questions are open for discussion. I would love to see some panel discussion as opposed to individual one-off responses. So let's kick it off with the question of looking across the entire field and not just your own work. What do you believe is the single most promising advance in synthetic biology to date? Jim, maybe I'll start with you, since you're at the far end.
So the single most promising advance, that's as I said, I had the mic off. That's an interesting question. I don't know. I mean, I guess it's probably the general framework that's introduced in that is that we can make biology programmable. And it's interesting, very simple in notion, but it's something that was really quite foreign to the field, to the biologists when we did this. And it really, when the field launched, it was basically a group of misfit toys that were bringing this engineering mindset, and it was not accepted by the biology world, but it's now become quite accepted. When you think of many of the advances that have kind of captured the imagination of biologists, it's around this idea you can now program living cells. And now maybe just as an additional historical point, which Ken will remember, is that in the 90s, there were major debates around the United States around the misuse of the term genetic engineering. I used it in my talk, but that the feeling was that the biologists and geneticists had co-opted the term without having any engineering. And I think it's really nice that synthetic biology has squarely brought engineering now into molecular biology.
Wonderful. Does anyone want to add? Mo?
Yeah, I can add a little bit to that as well. And so you asked about sort of what are the kind of successes or single most. But I'm actually gonna cheat.
I need to pin you down.
I'm gonna cheat a little bit. I'll give you an answer on a few things. One is product, tools, and just vision. And so along the lines of what Jim mentioned, I think one success, if you will, of synthetic biology is this very exciting kind of vision that has allowed for and energized a lot of young people to join the field, right? And I think we can call that a success, right? Just seeing the sort of how the field has really expanded. In terms of tools, in some part motivated by this vision, this has inspired a lot of people to just develop technology that has advanced things like writing and reading DNA, right? So like when I was in Jim's lab as a postdoc, I was doing old school cut and paste, molecular biology that looks nothing like what folks in our labs are doing now. And that's because as people have been motivated to sort of reprogram cells, this has pushed technology in like really amazing ways, I would say. So that's a kind of a bystander effect of synthetic biology. In terms of products, there's a lot to name. But in some ways, a lot of people actually champion and call or inherit the CAR T cell framework as really a big success. And that's because it's one of the first examples of using and engineering a living cell to have a major impact in the world, and in this case, in medicine. So in some ways, Wilson was way ahead of the game here, thinking about this.
And can I just ask, do you see that, with bringing more young people into the field, do you see its increased collaboration across disciplines? So now bioengineering is working more closely with some of the more traditional biological areas?
I kind of want to give this to Mark because
I appreciate that. That's very nice. I would say certainly. There's no doubt that the technologies, the way of thinking, the methodologies, the techniques, all of that is becoming a larger part of what I would call more classical areas of biomedical engineering. Drug delivery would be a classic example. Wilson and I have a nice project there. We're trying to actually spatially control some CAR T cell activity. And again, the marriage of those disciplines really opens up new and exciting projects. So I do think it has far-reaching consequences.
I think maybe to add to that, as a younger person on the panel, recently in grad school, I mean, as I was going through, I would say a bunch of projects would come up all the time where folks would reach out to Wilson, or Wilson would reach out to other people to work on more classical biological applications and incorporate synthetic biology elements in them to maybe read out of a novel assay or develop a technology that otherwise wouldn't have been possible. So I do think, especially at the trainee level and hopefully beyond, it is encouraging a ton of collaboration.
Fantastic. So looking ahead to the future, we're talking about successes now. This is a question I posed when we got together before. It's the crystal ball question. Looking ahead, maybe 5 years, 10 years, not necessarily 30 years, 5 or 10 years, what synthetic biology product or application do you expect to be in common use? Aside from, we're already making progress in CAR T. Do you see other areas? Mo, you mentioned agriculture. Do you see other spaces where synthetic biology could really make a difference? Anyway, yes, Mo.
Thanks, Jim. I do think, I do think it will have an impact in the area, I'll just call not just agriculture, but sustainability, more broadly speaking, because, what we're talking about here at a very high level is using biology, which is the ultimate renewable source, right, to produce, commodities, but also to produce really useful and valuable products that everybody here uses, eats, et cetera. And so the more we understand how these systems work, like plants, for example, and the better tools we have to more rapidly manipulate them to, for example, accelerate plant breeding, because, no surprise, the climate is changing very quickly and we need crops that are resilient, right? And synthetic biology is very much about accelerated, engineering of biological systems at the genetic level. And so, I see this hand in hand, and I feel like these are technologies that are going to be quite useful in sort of addressing these types of issues. I think we'll see more and more advances in the space of therapeutics and medicine and diagnostics beyond the CAR T cell. We're already seeing this from the work of this whole panel. I very much am excited, as I mentioned, about not just kind of engineering cells as medicines, but actually using technologies such as yeast, right, genetic circuits and directed evolution to design precision antibodies and biologics. And I think that that I think is sort of an indirect technology, angle in which synthetic biology is going to be quite useful in the space of medicine.
Thank you.
Yeah, I'll maybe add 2 areas. I think the first is, I expect in the next 5-10 years to increasingly see synthetic biology being used by molecular biologists as tools to probe the inner workings of living cells from a basic science standpoint. We've not had a big impact in that space yet as a field. I think most of us is focused on building our toy systems. I'll call it toy, I don't mean that negatively, but of small systems that we like, we analyze. But I think kind of increasingly using synthetic biology constructs, kind of like the voltage clamp equivalent in neuroscience. The second is, I think SynBio is going to play a major role in generative biology, AI, of providing functional data for AI models. So these models are, of course, data hungry, but more so they need data around function. We're swimming in sequence data. We have a good amount of structured data for protein, not much else. And I think you'll see synthetic biology advancing high throughput means, some automated, some not automated, that are going to feed models that are going to enable us to make biology more of an engineering discipline.
Thank you. So maybe we switch it up a bit now. So we'll move away from the science and move more towards where Wilson has also had a lot of accomplishment, and that is translating, moving the science out of the academic lab into industry. So each of you has had incredible experiences working with industry. You're either collaborating or in many cases you formed your own companies. So I'd like to ask you, and maybe I'll start with you, Justin, because your talk really focused a bit on this topic, is what is the difference between answering a question in an academic lab, a scientific question in an academic lab, versus addressing that same question in the industry setting in a company?
Yeah, so thank you. That's a great question. I think it's also a really important question because from an academic perspective, you're not always trying to do something with like super hardline focus on the endpoint of a product that you're trying to develop or a disease that you're trying to cure. A lot of the times it's much more exploratory, where you will set off to accomplish a goal, do a particular project with a collaborator, and along the way you'll have a lot of exciting findings and you'll want to turn over every single rock. And I think that's really important because it leads to a lot of groundbreaking science that otherwise wouldn't happen. Transitioning that though to like a more industrial sense, where on some level or another, you are beholden to investors and you are also beholden to public benefit, where you have sought out to do something and now you need to do it. Not saying that you're not turning over rocks along the way, but I think it's important to have a much more focused, targeted approach to what you're trying to do. And really making sure that the questions that you are asking about the system that you're trying to develop, about the drug that you're trying to get into the clinic, are directly addressing things that you need to solve before convincing yourself that what you're doing is going to work and what you're doing is going to have public benefit.
Anyone want to add to that?
I don't think I have anything useful to add except to. But I love talking. I'm gonna use it to shower praise on Wilson, actually, because it was Wilson who, over the years I would sort of go into Wilson's office and say, check out this genetic circuit we're thinking about. And it's like, that's really cool, but what the hell is that useful for? And I think, but I think I'm saying this, I'm joking, but like, he did kind of motivate us to think about mission-driven, exploratory work, is what I like to call it. There's this, famous thing called, Pasteur's quadrant. Do you guys know about this? Like, Pasteur was this amazing scientist who, could kind of balance both doing kind of basic science, but having a mission and like really trying to impact. And I think Wilson really sort of epitomizes that and really helped push us in that direction, to the point where, this is sort of kind of what pushed us to think about how we could use the basic science of genetic circuits to think about making new types of medicines, right? And I do agree with Justin that there's a difference somewhat in the sort of missions between industry and academia. But I think this era, I think the sort of vision, I think the sort of the pathway of doing stuff in academia, pausing and saying, all right, let's kick it to company, right, is flawed. And I think it has worked in the past, but it's not going to work, for the future. And I think there has to be some sort of like hybrid mission-driven academic science of collaboration between industry and, academia. We are facing a crisis in academia. Surprise, right? That I think will be really useful here. And I just want to say, I think, Wilson kind of epitomizes that, right?
Thank you. So speaking of industry and startup companies, which Wilson has been part of 3, right, startup companies, if I could ask the panelists, those who have been part of a startup company, what was the biggest misconception you had at the time that you started the company? And I'm going to follow that with, what piece of advice do you wish someone had given you before you embarked on that journey? Mark, I'm going to single you out there because I see you contemplating these questions.
I should have taken the easy question. Serves me
Remember,
right.
you could always pass if you have to.
Just 2 or 3 things? Okay.
Do that.
Again, I think there's a lot of things that go through your mind when you're starting to think about doing a startup. And I think by far being extremely critical of your own work as well as other work out there is essential to think about, okay, is this worth starting a company or not? What experiments do you need to do now that would give you some justification for putting in money? And that means literally your own money, right? So, are you willing to put money into it and pay for someone to work in the company? And if the answer to that question is no, then you're not ready to do it. And so you have to be extremely critical about the science. You need to think about working with a group of people that you know you can communicate with, talk with, and just be very honest about the science and where you're going. And I think for me, that's been one of the pleasures of working with Wilson. Incredibly critical, always asking questions, always pushing us to think about what to do next and why to do it. And for me, I always think about companies. You don't start a project to do a company. You start a project because you have an interesting question you want to do. If the technology and the science takes you there, then you keep pushing forward. And when the time is right, then you do it. So to me, I think it really is about the science and building the team. And I would say those are the things that I think about the most when I'm doing a company.
Maybe add a few comments. So I've had a number of companies spun out of my lab. I've never had an operating role in a company, so it's more of advisor looking in. But a few comments, particularly for the young folks. One is, I think it's really important to know what you know and know what you don't know. And that for the most part, when you're trained as a grad student, you're trained to do really great science. And that often does not translate well, pun intended, into work in a company. Second is that be really careful who you team up with. That it's easy to form collaborations in academia. It's easy to get out of a collaboration in academia. It is not easy to get out of a team you create in a company. There's legal binding and sorts. And I've seen a lot of young people make mistakes and not their due diligence on who they team up with. And then three is that if for a company, it's not simply a place to do great science. It needs to have market pull. You need to be producing somebody that somebody wants to buy. And just because you publish really cool science in a Nature Sciences Cell paper doesn't mean it's a company. But it seems like every young person in this city and the city across the river think that's the defining feature of a company, and that's really wrong. And you'll be able to start your company. There's almost no barrier to starting your company, but it's really hard to create a good company, let alone a great company.
Thank you. So one last question, and I'm hoping each of you might be willing to share your thoughts on this. So we're here celebrating Wilson Wong, Innovator of the Year. So, and your contributions have been, again, scientific as well as in the translation space. I'd like to ask the panelists, based on your own experiences, what does it take to be innovative in science, as opposed to just productive or excellent? Justin,
I'll take
you'll
the
be
first
brave.
stab. Obviously, this is not the be-all, end-all. There are a lot of answers to this question. I think one of the things it takes to be innovative, though, is to surround yourself by a lot of smart people who know a lot of things that you don't. Whether that's through collaborations, whether that's through building a team at a startup. A diversity of experiences, a diversity of backgrounds, and experts in their field, again, that you are not in, leads to a lot of conversation and a lot of ideas that you as an individual would never have, and that other people that you're working with may never have because they're not benefiting from your expertise or experience. So I think that to me is like a hallmark of innovation. And I think Wilson does a great job of that. I mean, I feel like when I was in your lab, there was a new collaboration almost every week. And it was exciting. It was really fun to be a part of.
Thank you.
I just thought of just one thing, and then I'll give it back to you, don't worry. I think trying to have an open mind and not judging too quickly, and looking at the experimental data. So sometimes when experiment doesn't work, that's actually more interesting than when it does work. So I think, kind of keeping that mindset and not being, it's very easy to get tracked and only think about one thing and never look up. And I think it's important to try to do that, but that's super hard to do.
I'm just going to echo a few things that have been said. At least for me, I've really enjoyed working, and this is building off of Justin's comments. I've really enjoyed, learning from others and building, interesting teams within our lab and trying sort of experiments that way, and that's worked quite well. Some people look at our research portfolio, which spans everything from bacteria to yeast to plants to immune cells, and think we're crazy. I think it's normal. I think it's interesting and fun and an interesting environment for people to come and do, science in, and that doesn't bother me. But, in order to sort of kind of be innovative across those types of areas, you need to sort of foster really good collaborations, and you need like support structures within the groups. And I think that's what we've done a really good job of. And then the second thing actually builds on what Mark said. Yes, it's important to fail fast, but it is also important to sort of be patient with the science and like, have some conviction about, big ideas and don't give up on them. I probably take that a little too far with some projects going 5-6 years until we still have figure one done, right? And so maybe that's an extreme, but sometimes it's like a good wine. You open it, you let it breathe for a while, and that it just needs a little bit of that time and patience to really come to fruition. So, yeah, if you have conviction, take your time with it.
Yeah, I'll answer with Wilson in mind and give 3 features that I think describes Wilson but also captures innovative spirit. One is, I think it's important to be really a deep expert in a technical area and deeply familiar with the content of your field, so you can be rich in many ideas and see their context. Second, I think you need to be respectfully irreverent in that, not to just take what people give you, but, challenge what they give you. And 3 is you put in a lot of hours in the lab and at your desk. Wilson's brilliant, but Wilson knows that most of his good ideas aren't going to work. And he gets in there and he's willing to fail fast, but he puts in the time. And having that rich technical, knowing where it sits and being irreverent about himself as well, has enabled him to make major advances really quickly.
Wonderful. So on that note, I'd like to thank our panelists, Justin, Mo, Mark, and Jim. Thank you for your insight. And again, we want to congratulate Wilson. Well deserved.
Good afternoon, everybody. I was going to introduce myself, but Jim already introduced me. So I have some remarks here about Wilson, but I'm not going to start with them. I'm going to start with a little bit of a historical perspective of how we got here, and then talk about Wilson specifically. And I want to do this for a variety of reasons, particularly given the panelists and the people in the room. Most of you who know me know that I'm deeply passionate about the value proposition to society that our leading research universities have. The innovation ecosystem begins at our great universities, and even though it's currently under systemic threat, it's been an incredibly successful one over the years. And it works because we leverage the creative science and the advancements of new understandings that can come out of research at universities. But that alone isn't enough. It actually then leads to people at these institutions that see the light of maybe this can lead to a new innovation that can change people's lives in a very positive way. Obviously, particularly this is relevant in healthcare and in the medical fields. And so that's an enormous value to society. And we deeply value that in the way and the kinds of faculty we recruit and the kind of students we train and postdocs that we train. I'm going to give you a little history lesson. Back in a lot of years ago, I was chair of biomedical engineering, And we made a decision that we were going to try to become one of the best bioengineering departments in the country and one of the biggest. And we competed for a huge, at that time, about a $16 million grant to grow the department to be the largest and most diverse in the nation, with the fundamental goal of connecting engineering principles at the molecular level, biomolecular level, to the cell level, all the way up to the organ level. And Jim was a major leader of the three key themes at that genetic level, at synthetic biology level. And we won that grant, one of only 100, of the 100 applicants, we were the only program to win that grant. And then we started to recruit faculty. We had about 13 faculty, but we recruited a different kind of faculty. We recruited faculty who were not just basic scientists, but with a clear eye on how to use their research to translate it into new ways that improve people's lives. One of the first recruits from that was Mark Grindstad, he was at Duke University at the time. And Mark came in and didn't just do chemistry or chemical engineering or biochemical engineering. He was clearly translational. He was driven to come up with new ideas that would improve people's lives, start companies, some of the earlier work on lubricants, the chemical lubricants, polymers for lubricants, and so on. And we recruited other faculty just like that, Ed Damiano with the bionic pancreas, Kathy Clapperidge with the precision sensing, and so on and so forth. And it started to change the culture that we can do great science, leading to great innovative ideas, and then translating them out through support of going out to those new companies and getting products out in the market. Then I became Dean of Engineering, and it was that by that point, Jim had really identified BU as the origin of synthetic biology as a field. And he was really putting a huge sign on BU as we were changing the world through synthetic biology. And we had a grand total of one faculty doing that, and that was Jim Collins. And so I realized, and Jim and I had a lot of conversations, we got to grow this group and leverage this reputation that we've got. And so we have to add some more faculty beyond the ones we recruited with the Whitaker Foundation. And so I said, well, we're going to need some money for that. And of course, we both realized that the real stars were really emerging. It was a very young field. And I remember walking into Bob Brown, the past president of BU, and in there was also David Campbell, who was the provost at the time, and Andre Rukenstein was the vice president for research. And I asked him for $5 million to recruit several new faculty in synthetic biology, junior faculty. Bob proceeded to scream and yell at me how greedy I was and how selfish I was and who do I think I was, taking all the money the university had, that I was the most, who thought that engineering was the most important thing, and biomedical engineering. And I was just stunned by this. And he ended the conversation by saying, I'm going to give you $4 million and that's it. and it was like you could hear a pin drop, and I just looked at David Campbell. He goes, leave, get out, get out, get out. And I walked out. And Andre and David said, that's the most money I've ever seen him give away to anybody. And we used that money incredibly well because we recruited Mo Khalil and Wilson Wong. And we had the trio, the Mo, Larry, and Curly of synthetic biology. And we, and I had to do the picture up there. And that had just ramped us up on the map as we were doing great things with understanding how to manipulate biomolecules, genetic circuits, cells, and then translate them out. And Mo and Wilson got the same bug that Mark and Jim had. It's about not just basic research, not creative, not just intellectually fearless research, but started to see, hey, this could actually improve people's lives. And they embraced translation, and we've got to do that more across the whole university. So that's why I'm excited today to introduce the winner of the Innovator of the Year Award in Wilson Wong. And now you'll indulge me as I read the script because I think it well describes the background we have here. We're talking about a professor who's a leading innovator in the field of biomedical engineering and synthetic biology, serving as a faculty member here in the Department of Biomedical Engineering, and he leads research at his lab with in the university's Biological Design Center, where his work focuses on developing advanced strategies to regulate gene expression and control cell behavior. And by integrating engineering principles with biological network design, molecular biology, and chemical biology, Dr. Wong is pioneering new approaches to medical treatment, including the next generation of CAR T-cell therapies. His research is central to the development of safer, more precise gene-based medicines with broad applications in vaccines, immunotherapy, protein replacement therapies, and cell-based treatments. Wilson's work addresses a critical gap in modern biology. While our ability to observe biological systems has rapidly advanced, our capacity to dynamically control them, particularly in complex multicellular organisms, has lagged behind. Over the past decade, his lab has developed sophisticated tools to regulate mammalian cell function at multiple levels. These innovations enable precise, independent control of multiple genes and cellular processes simultaneously, offering unprecedented capabilities for both scientific discovery and therapeutic development. His systems are designed for robustness and versatility, and have been validated in preclinical models, including in mice and primary human immune cells. A major focus of his work is the application in immune cells for therapeutic and diagnostic purposes. His lab advanced the logic-gated CAR-T, has been pointed out several times, CAR-T cell therapies to improve tumor specificity, as well as engineering universal CAR-T cells to target autoimmune diseases such as type 1 diabetes. Additionally, his work is constructing multicellular circuits that is shedding light on complex biological processes such as lung branching morphogenesis, with implications for regenerative medicine and tissue engineering, linking him to a former Innovator of the Year Award, Darrell Cotton, who's sitting in the audience, and the director of the Center for Regenerative Medicine. He's a prolific inventor and scholar. He has 13 issued U.S. patents and many pending applications across 14 patent families. His collaborative work spans both academia and industry, including notable partnerships with the Greenstaff Lab, as you've heard, leading to the development of self-amplifying RNA, a platform for delivering genetically encoded therapeutics. This technology formed the foundation of Helicon, a Boston University spin-off company co-founded by Dr. Wong, focusing on durable RNA vaccines and therapies, co-funded with Justin and with Mark, and others, maybe. He's also co-founder of 4Immune, which develops therapies for autoimmune disorders, and Senti Biosciences, as Jim pointed out, a clinical stage biotechnology company advancing innovative treatments for cancer and other serious diseases. Now, if you know Wilson or you've never met Wilson, when you do meet him, you'll say, this guy's kind of quiet. He's kind of unassuming. And then if you have him give a talk describing what he's doing research-wise, or ask him to talk in front of a group that you invite him to be a speaker on, first, every time you do it, you forget, oh, he's kind of quiet and unassuming. And then within a few minutes, you realize, oh, but he's really substantive, and he's really creative, and he's really intellectually fearless, and he's brilliant, and he embraces the power of convergent science across multiple disciplines to think about creative solutions to really important problems. And he's thinking about translating it out to the real world. So this is every dimension you need in an enormous Innovator of the Year faculty member. He's also, of course, by doing that, mentoring new generations of students and postdocs that are going to be translational and recognize the power and need to do that in the academic community. His contributions to science and innovation have been widely recognized. His honors include, he's an Allen Distinguished Investigator. He's won the National Science Foundation Career Award, the NIH Director's New Innovator Award, and multiple BU Ignition Awards. He's also received the ACS Synthetic Biology Young Investigator Award and the BU College of Engineering Early Career Research Excellence Award, among others. Through his pioneering research, entrepreneurial leadership, and commitment to advancing human health, Professor Wilson Wong exemplifies the spirit of innovation and impact recognized by the Innovator of the Year Award. Wilson, congratulations, and I've got this giant thing to give you. You don't have to say any words at all. It's your award. Okay. Just run off with it. We'll just sit and talk.
Thank you so much. I must say this is weird. Well, first, I would like to thank the Golden Globe Foundations and my agents. Sorry, when I get nervous, I like to joke a lot. sometimes it's inappropriate. So, well, first, I do want to say, well, for Ken mentioned, I guess when I was younger, I used to be very arrogant. I mean, I still am. But my wife and life have beaten out of me a little bit, so I try to be a little bit more modest. So, but well, first, thank you for Ken and OTD for giving me this award. It's a huge honor. I know many people who have previously won the award, from Jim, Mark, Daryl. I mean, there's many. I can name them all. They were all inspirations to me. I must say, I looked at the list for a long time. I wish I was going to be one of them. So thank you. So this is me being arrogant. I'm sorry. Yeah, it's overwhelming. I've been to, I listened to a lot of these talks before, never about me. So again, so thank you. I was very nervous when I learned, like, Jim, Mo, Mark, and Justin's going to all say something nice about me. I know it's difficult. I know it's torturing. So I'm glad you made it through. So, of course, I'm not going to talk about innovation, not in front of everybody here, or my research, and I feel like I'm not qualified to do that. But I do want to thank a lot of people. First, I'll just start with the panel. I want to thank Jim. It's like, as you all know, he's the pioneer of synthetic biology. When I first started graduate school, when I was at UCLA, joining Jim Liao's lab, the first paper he showed me was your work, Jim. Instead of his lab's work, he showed me, you gotta read this paper, the toggle switch. And I was like, what the hell was that? It's mind-blowing to me at 2001 when I was reading that paper, toggle switch. It's the whole reason why I'm doing synthetic biology. You are the reason why I come to BU. It's 36 Cummington. Like BU, to me, it's important to me. 36 Cummington, it's a historical landmark to me. I know we talk about tearing it down and things like that. We gotta save that bench. I don't know which bench, Tim Garner made the first toggle switch. We gotta save that bench. Don't tear it down. If you tear it down, give me that bench. I'll find someplace. So anyway, so Jim, having you say something nice about me, I still remember when I interviewed with Jim, and then he's very nice. He was like, I was like, oh, I don't know what the hell I'm doing. I'm very nervous, and then Jim was like, oh, you're doing great work. But one thing that stuck with me is like, well, you gotta hustle. Hustle. I was thinking to myself, I've never used that word in my life. I'm from California. I don't hustle. I feel like, right? Is that an East Coast thing? But that stuck with me, right? A long time. I still tell my students, like, you gotta hustle now. You gotta work hard, right? So yeah. Anyway, Jim, you're a huge inspiration for me. Mo, it's difficult because, we come up together. So as I'm really happy, as a friend, see you doing great things and all of these things. Of course, I'm sad that you're leaving. But, I have plans for us. I'm like, if one day I don't do this work, I'd like to be comedians. My wife thinks I'm not funny, but I'm going to try anyway. So I have plans, Mo. You and me is going to become, like back then when we first started, Mo, not knowing, we're in this comedic bit, right? I always say I'm Mo's secretary because we share an office. And then I always sit next close to the door, and then people always ask, like, where's Mo? I'm like, I don't know. So I'm like, so I always joke around. I'm like, I'm gonna get Mo's coffee. I'm gonna get his dry cleaning, and things like that. But so here's what I'm thinking. Okay, I already have a plan for us for retirement. Okay, so we're gonna start this comedy duo like like Steve Martin and Marty Short. Okay, we're gonna have a show instead of Only Murders in the Building. I like it a lot. We'll have Only Salvatore in Silsey. Every season, we're going to have trying to figure out who sabotaged someone else's experiment. Sorry, Mo. See, this is me testing out my jokes. It's not quite working, so I'll So Mo, I love you. You in a lot of ways, I feel like you're a much better professor than I am as a professor. I'm always looking at your lab and trying to like, ooh, Mo got some cool research going. Can I use it? Can I put them in T-cell? But anyway, I always look for you. So, what would Mo do? And my students know. I'm like, if you want to write good paper, read Mo's papers. He's like really good at defining problems and then executing on them. And then Mark, I want to say thank you. I feel like the last few years you have adopted me in some way. I still remember, I work closely with Mark. Mark works really fast. I can't keep up. He writes fast. He thinks fast. It's like I really can't keep up sometimes. It's like I gotta work like twice as hard just to keep up with Mark. And he's a fantastic, coming up with ideas and executing on them, and also an excellent grant writer. I still remember year 10, I wrote some grants, and then Mark told, I was being lazy in my summary and project statement, and I would send it to Mark. Mark sent it back to me, he's like, this is not good. And then I'm like, okay, I got a lot to learn. So over the years, not just in science, but also entrepreneurship, Mark have obviously, many of you know, have founded many companies, have a lot of successes. It's a different way of thinking, if you get buy Mark more wines. All right, you'll learn a lot about finding companies and how to exit and things like that. So I'm still learning from you, Mark, and thank you for working with me and teaching me all these things. So really appreciate it. And then Justin, thank you for coming back. Having you've been a great student in my lab, and talk about fail safe, fail fast, and try. This is something that you also I try to share that with you. Not everybody internalize it. Okay, you're some of the students who really take my BS in some way, and I know you don't have to come back, so I take it as a compliment that you didn't hate me enough, that you're still willing to come back, so appreciate it. So also want to thank, none of the work that we talked about, in my lab, I didn't do any of the work, right? I don't even know where the pipette is. I tried to do some work in the lab last year, remember? My trainee got a little nervous. They were like, "Is the funding so bad?" Right? So none of these work, of course, would have happened without the amazing trainees that we have at BU. And then also, I do want to say a little bit about the environment at BU. I always feel, BU, it's incredibly nurturing for me. Life is pretty difficult. Science is getting harder and harder to do these days. But I know within the BU community, I have friends. It's very collegial. It's one of the most friendly place I've ever been to. And you can talk to anybody. Everybody is very respectful and very nice. It's so, for people like me, I feel like I can't deal with politics. So it's such a nice environment. It's kind of like having a protected home here. And that really helps me to be more creative, helps me, willing to take more risk. So I love BU, I love the environment, not just because you gave me an award, but this is it's an incredible place. It's with amazing colleagues, so I really, really appreciate Ken and Jim taking a chance to hire me back then. I still remember engineering T-cell, and that was 2010. It wasn't a thing, No one really know what to do with it. So I like to think, Jim, you saw something in me that people didn't know back then. I think you saw the potential of immune cell engineering that many people didn't recognize. So, and then, of course, Ken, willing to hire me, so I really, really appreciate it. So, that's all I have to say. So thank you very much. I hope you enjoy the day. Thank you.
Thank you, Wilson. That was excellent. I invite you all to a reception now and mix and mingle and perhaps congratulate Wilson and get a chance to say congratulations. Thank you, everyone. Thank you for the panelists as well. Thank you very much.